Thank you very much for the introduction. So, hello everyone, my name is Tong Li. I'm a senior software developer working at the Wellcome Sanger Institute, working in Omar Bayraktar's lab. Today I will be talking about the WebAtlas pipeline and for integrated single cell and spatial spatial transcriptomic data. So as you may know, Wellcome Sanger Institute is well known for its work on the Human Genome Project that was finished 20 years ago. And recently, led by Sarah Teichmann, we're also interested in spatial transcriptomic data, basically the imaging related sequencing data. And among the Human Cell Atlas, in the consortium called Human Cell Atlas, Sarah that initiated, we have one project called Whole Embryo Project, which we are looking at here on the left. We have human embryos taken from the donors, and we just want to apply different technologies onto it, including single-cell RNA-seq, Visium, and Xenium. The reason we want to apply all these different technologies is simply because they are complementary to each other. So, for example, for single-cell RNA-seq, you don't have actually the spatial information attached to it, even though you have the whole transcriptome profile of each single cell. But as with the spatial transcriptomic data, so the IST, either by imaging-based IST or sequencing-based IST, you actually have the spatial information attached to each single cell, and you can combine it with the sequencing. But they still have their own shortcomings. For example, the image-based IST, you can have true single-cell resolution because you can do single-cell segmentation, but you can only you are limited to 100 to 1,000 genes per cell because of the constraint of the technique. But if you look at the sequencing-based ST, you can actually have the whole transcriptome-wide profiling, but the resolution is limited because it is limited by the size of the Visium spots, for example, in this case. So it will be ideal to combine these different modalities together, so in order to get a holistic view of each single cell in human body. So the idea is to actually create a Google map kind of thing for all human cells. But actually, we do have some technical challenges. The first, as people here are very familiar with, is the data format issues, especially for imaging. But and so it is started to be unified, but not that web friendly yet. So the reason I mentioned web friendly is because the second challenge, because we are looking at something that is easy to use for browse multiple modalities, meaning that we don't want the user to install anything on their computer. So we just send them a URL, they click on it, they will be able to browse the data. So in order to tackle these questions here, one thing I should mention, if you're familiar with the spatial technologies, you will know that spatial data is coming out as a unified data formats for all the spatial transcriptomic and single cell data sets. But this project and WebAtlas emerged almost at the same time. And so our pipeline is not fully adapted to spatial data, but we are actively talking to each other to make sure that we'll be using the unified data format. So here is a WebAtlas pipeline that can potentially facilitate spatial data dissemination across the world. So the idea is pretty simple. You just take different data sets from different technologies, either in TIFF files format or H5AD files for your tabular data for single cell analysis. You pass them through the Nextflow pipeline that we just wrapped everything into the pipeline so that you can do the conversions, and you don't need to care about the environment setting, etc. Don't need to care about Conda or Docker images. And then what you need to do is to define your parameter files as in the middle. So the most important part is that you give the path to the end data object, pass to your raw images, and pass to your label images. And then the pipeline will take care about the conversions and building a WebAtlas visualization config file so that it is ready to be visualized on WebAtlas. WebAtlas actually, if you're not familiar with if you don't know what it is a work from a Neil Schellenberg team in Harvard Medical School. They built it for visualizing spatial genomics data on the web portal. So we just leverage that framework in WebAtlas. So in order to run WebAtlas, it's just a one-liner. You just do Nextflow run, pass to the GitHub repo, and your parameter file that you configure in the middle. That's all you need to do to prepare your data to be ready to be visualized on the portal. And if you want multimodal data set to be visualized on the portal, you just add a second step again, Nextflow run. And everything will be ready for you as long as you put them onto the S3 buckets. And we also had a sort of rough documentation of how to do that. If you want, just have a look. It is on our website, so feel free to reach out. So actually, even with all the one-liners and the documentations, the reviewer of Nature Method still think it's not user-friendly enough. So we came out with this idea that, okay, what about let's build a portal. User can just drag and drop data set into it, and it will run automatically on using our Sanger services at this moment, Sanger storage. And then by the end of 10 minutes, depending on the size of the image, because the conversion does take a little bit of time. So after 10 minutes, you will be able to receive a link, and you click on it, you'll be able to see whatever your data set you submitted to it. This is a work in progress done by Martin Preet, which is our Celgene IT lead. So I'll be showing you a live demo about WebAtlas. Before that, I want to show you, when I click on this link, you'll be able to see all the data set, actually all the figures that we used in the paper. And then if you just want to see what the data actually looks like, you can just click on the link to visualize it. One thing I want to mention is that we have, for figure 2A, we've also submitted our data to Bioimage Archive, so that actually we're directly digesting the data from CBIAS S3 buckets. So we are not hosting the data at all. So I will do the live demo. Hopefully you're not seeing it, sorry. Do I have to quit the full screen? I must be doing something wrong. Yeah, okay, I'm here, thank you. So as you can see, I click on the link and you have a web portal. This is abstract of the paper. And I just click on the WebAtlas. And here you're looking at integrated multimodal visualization. On the leftmost is a single cell RNA-seq. In the middle, you have in situ sequencing. And on the right, you have a Visium datasets. For people who are familiar with the test, this is a very familiar view for you. If you have used it before, it's nothing really different. You can zoom in and out. Everything's raw data, because if you zoom in, the highest resolution will be automatically loaded. So new feature that we introduced into this portal is that actually you can find for different genes. Let me zoom out. And if you click onto that gene, all the different modalities will be automatically highlighted. So basically, we will be able to look at the expression of these single genes across different modalities. And the same concept applies to cell type annotation as well. If I click on control site, we will see the cells in different areas will be highlighted. And if you find something interesting and you want to annotate, you can also do that just using the lasso tool. Sorry. You can just draw some polygons, and you will see on the right, the annotation will be here. And you can visualize them and then download them as either CSV files or JSON file. This is actually the way that we are using this tool in our project. So this part is done by Sid Lawrence, which is our clinical PhD student. He used the portal that I sent him. He annotated some regions which is supposed to present some pseudo-temporal differences between different regions of a developing human limb. And actually, this enables the possibility to just see how quantitatively the cell type evolves across different regions of the human embryo limb. For example, in the tibula, this phase, this is supposed to have relatively older cells, more mature cells compared to the progenitors. And in fibula, you should expecting a little bit less mature cells. And in the old ages, you should have the least amount of mature cells. And if you look at the plot on the right, actually, this is exactly what we're expecting. You have more mature cells on the bottom and a little bit less in the middle, and then the least on the top. And this is the first time that we can actually do this type of quantitative cell type analysis in different regions using an interactive tool that enables this possibility. So as I briefly mentioned, the whole purpose of this project to make sure that we have a web portal that we can disseminate the human embryo project data. And for that, we will be having 55 sections of Visium data sets. And we will want to build on top of this something that is browsable, like Allen Cell Institute. You can have just slider on the bottom. You can click onto it and to look at different regions, different sections of the embryo. And this part is led by Daniela and Dave from Mass Hanifas lab. And the last part is the image analysis in 3D. Actually, it's great that we already seen some point cloud registration this morning. So basically, we will be applying the same concept to the 3D in situ sequencing datasets. And we were thinking about using point cloud registration for 3D stitching, but also the registration of different imaging cycles for example, for in situ sequencing. And for that, please check out Stanislaw's poster, which is number 22. By the way, he chose this picture, it's not me. And in addition to analysis, and we are also working on, well, Stas was also working on visualization of the 3D dataset. Here, what we are looking at is the mouse brain 3D volume data, and which he was playing with VSPy, and you can automatically generate, just load the data into the RAM and visualize different angles from different resolutions. So one thing, so everything is served from Omizar, so it will be a scalable solution. And last thing, as you can imagine, this piece of work requires a little army to be able to enable it. So, and thank you everyone for contributing to this project, and especially David and Daniela. Actually, they're also the co-first author of this paper. And one last thing, we're also hiring, so stay tuned if you want to join Sanger. Thank you very much.
Thank you very much. Maybe we have time for one question, if someone has a burning desire to ask a question. Okay, then let's thank the speaker again.